Hello, everybody, and welcome to our webinar on optimizing nuclei extraction and counting for single-cell sequencing. My name is Andy Jones, and just before we get started today, I'll give you a quick introduction to our company for those of you who aren't familiar with us. So DeNovix was founded in 2012 in Wilmington, Delaware. And that's where we continue to develop and manufacture all of our instruments and software. Our products are based on our core expertise of high-end optics and detection technologies and integrating those into innovative products for the life science market. You might be familiar with the DS11 series of microvolume spectrophotometers and fluorometers. And more recently, we introduced the CellDrop Automated Cell Counter, which allows us to remove disposable plastic slides from the cell and nuclei counting workflows. And that's the instrument that will form the core of our counting part of this webinar today. So our focus has always been on providing the best in industry support. And you'll see on the right-hand side a number of awards that we've collected through the years based on product innovation, customer reviews, and our customer service. So today's speaker is going to be Ben Capozzoli, and Ben is a senior member of our application support team here at DeNovix. Ben graduated from Villanova University with a master's in immunology and brings a wealth of experience to our team in immunology, imaging, cell biology, and cell counting. So at this point, I'll hand over to Ben, who's in our lab in Wilmington.
All right, thanks, Andy. So today I'd just like to start off with the topics we're going to cover. So we'll start with tips for optimizing your nuclei extraction protocol. So we'll start with what kind of tissue you're working with, how to get that initially set up, and then how to actually go through the extraction so that you can optimize the yields and the efficiency of your extraction as you do it. Once we've completed the extraction part, we will actually move to a real-time demo. So I actually have a nuclei sample here that I will load onto the CellDrop automated series of cell counters and count here for you and show you all the tips and tricks to analyze the sample on the CellDrop with all the analysis tools that are built into the software. And then after that, we will move on to a Q&A session with myself and some of the other application scientists that are on the panel today. Before we get there, let's start with a poll question. So I would like to understand what types of tissues you analyze. So that could be fresh tissue taken directly out of, an animal. There's frozen, frozen could be, one of those preserved tissues, like a fresh that you preserve through freezing. There's FFPE preserved tissues. Of those options, do you use human tissue? Do you use non-human mammal tissue? Do you use plant tissue? I think we've conceived of everything there for possibilities in terms of nuclei extraction. So I'd just like to get a feel for what our audience today generally works with. Looks like a few more results kind of creeping in here. So I'll give everyone a few more seconds to finish answering and close the poll up now. So it looks like we have a heavy bias towards fresh tissue, but we have a good representation of frozen and FFP preserved as well. And it looks like we have a lot of representation on human and non-human mammal, as well as some plant people. So it looks like we got a little bit of everybody today. So hopefully I can answer everyone's questions, but if not, we have an opportunity to get to that later. So as just an overview of what we're going to do today, our webinar map, if you will. So we'll start with tissue prep. So preparing your tissue, whether it's fresh or FFP preserved or frozen, getting that ready for the actual extraction process. Then we'll move to the overview of the extraction. Once the nuclei sample has been extracted, so we have isolated our nuclei, we will walk through preparing that sample for quality control analysis on the CellDrop. After that, I will show you the counting protocol, so all the different settings that the CellDrop can use to best quantify the nuclei to your expectations. We'll talk a bit about imaging and nuclei counting and the different ways to do that and what the CellDrop can offer there. Then for the analysis portion, I will actually do the live demo. I'll show you how to count a sample and analyze it with the powerful, intuitive tools in the CellDrop software. And lastly, we'll cover downstream applications. So now that you have your nuclei, what are you going to do with them? I'd like to point out that throughout the webinar, if you have any questions, please submit them to our panel. We have some application scientists on standby on the panel to answer them as they're submitted, but I also will plan to answer a number of questions at the end of the session as well. So we'll see. But with that, let's dive right in. Let's start our journey at our tissue preparation step. So as we've kind of talked about a little bit already and alluded to with our poll question, you want to prepare your tissue, whether it's fresh, frozen, or FFPE fixed, to analyze it appropriately. So, we have a lot of options that we've worked with. We've worked with human, rat, mouse. We've had customers that have worked with tomato root and corn and other samples, and just a whole variety of different sample types that can be utilized. Some considerations to take here are if you have a really big tissue, even if you're extracting or grinding it up all in one spot, it might be beneficial to section that large tissue into several smaller pieces to go into the same tube to help it more efficiently extract. But your protocol will cover that in detail. So now that we've prepared our tissue, we will move on to the extraction phase. So this is where we're actually going to isolate our nuclei from the tissue. We have personally worked with the Nuclei extraction buffer and GentleMax system. So the GentleMax has been great for us because it automates the actual grinding process. So something that I would have done manually years ago in the lab, this equipment does for you, and they provide all the buffers and filters. So that's been a great way to process a lot of samples simultaneously. The 10x Genomics kit is another kit that we have used. It's a more manual kit that involves the manual grinding, but it's still an excellent extraction system. We have some images later in the talk that I can show that demonstrate how efficient that is on the CellDrop. And lastly, we have our user formulated buffer option. So there's, that's something that we generated in the lab here, just looking at stuff that was in the literature. And that also has worked really well. So the nuclei can be extracted in a number of ways. This probably doesn't even cover all the different options, but these are 3 that we've tried personally, and they've all worked really well on the CellDrop. So I have a montage that'll play as I kind of describe what's going on here for you of the nuclei extraction process. So you can see here that we're using the Nuclei GentleMax system. And right here in the video, I just put a cooling sleeve on top of one of my samples. You can see the system is doing the grinding for me, which really helps to make sure that all my samples are processed the same way, and one isn't processed differently than another. But the importance of that cooling sleeve that I put on in the video is one of the most important considerations in the nuclei workflow is keeping everything cold. So the nuclei are protected generally by the cell membrane, and when we isolate them, we release them from that cell, and what happens is they're a little more fragile. So we want to move quickly in this process, and we want to keep everything cold to slow or prevent osmotic lysis of the nuclei. We want as many of these to make it through to the sequencing step as possible. The best way to do that is through cold and also working quickly. To help you work quickly, I advise that you prep everything well in advance. All your dilutions should be done way ahead of time, and you want to make sure all your buffers are chilled or warmed to the appropriate temperatures according to the instructions for the kit, to be sure that you don't have any wait time in your protocol, because that will affect your extraction efficiency if you're pausing to dilute something. And then lastly, a couple of the steps in this video montage that you've seen, I've been pouring samples through filters. That's really important to understand what is going on with your tissue and what filtration steps are going to be needed to remove as much cellular debris as possible. We don't want a lot of debris when we get to the sequencing phase, so removing that debris beforehand is important. And different tissues might require a different pore size on the filter. So that's just, those things you want to consider here. So with our extraction, our tips here on the extraction completed, we will move on to talking about prepping our isolated nuclei sample. So we now have our suspension of nuclei. We want to prep it to count for quality control analysis on the CellDrop. So the first step here is equilibrating your AOPI to room temperature. Definitely keep the nuclei suspension on ice. We want those to remain cold, as I noted before, to ensure that they are stable. But the AOPI will perform better when it's warm to room temperature. Like all fluorophores, the cold can kind of quench some of the signal. So we want to make sure that there's no signal loss from just being cold. Once the AOPI has sat out at room temperature for a few minutes, we will add that to label our nuclei in suspension. So a 1-to-1 mix, similar to trypan blue method. So you'll just take, in a fresh tube, you'll put a small volume of your nuclei isolation and then an equal small volume of the AOPI and pipette up and down a few times to mix. Before you move to the counting procedure on the CellDrop, if you maybe set the tube down to do something and come back once it's been in the AOPI, it's really important that they are thoroughly mixed so that everything is homogeneously resuspended before it's loaded onto the CellDrop. I would like to point out that there is no incubation time required for AOPI, so it's not something you have to rock for 5 minutes or anything like that. It's simply mix 1-to-1 and input on the CellDrop. There's no wait time needed. I would like to point out, I have been talking a lot about AOPI, but trypan blue is something that can be used to count and quantify your nuclei. But the combination of AOPI and the fluorescent signal of the two stains removes a lot of subjectivity from our counting and offers a superior accuracy in the count. So trypan blue does work. We just strongly recommend the AOPI considering the nature of the sample here. So again, I'll show you another brief montage of prepping our sample here. As I've pointed out, the first tip is making sure that your AOPI is appropriately warm to room temperature. It doesn't take too long, just a few minutes, but it is important to do. It's ideal to minimize your volume of nuclei counted. A lot of times these don't yield a lot of sample volume, so you don't want to take 10% of your 20% of your sample just so you can do a nuclei quantification. So we actually have options on the CellDrop where you can use less than 10 microliters of your nuclei sample to make sure you maximize how much you're inputting on your final analysis. When you're resuspending the nuclei, you want to be thorough, but you also want to be delicate. The nuclei, as I mentioned previously, are very fragile. So if you vortex them too heavily, you can pop them. I typically recommend just pipetting up and down a few times or very gently vortexing. And then once everything's mixed with the AOPI, I recommend moving quickly so that you can get the sample processed quickly and move on to all your other various samples and then your sequencing as fast as possible to minimize osmotic lysis. So now, before we get into the actual quantification, I'd like to give you a little bit of information about the star of the show today, the CellDrop, that allows us to count these nuclei or cells without slides. So the CellDrop is a precise and reliable nuclei quantification method, and it's critical for QC in this extraction workflow. It's an automated counter, so it's an image-based automated counter, and it has no consumable required. And it can take advantage of these fluorescent AOPI labels. It's to provide fast, reliable, and precise nuclei counts every single time. There's a few key features of the CellDrop I'd like to cover. Most importantly is that direct pipette. So direct pipette, as I mentioned, requires no plastic slides, so there's no plastic consumable. That's great for any green initiatives at your institution. And it's also great for your budget, and you don't have to worry about remembering to have another piece of plastic in stock to do your actual experiment. But the direct pipette offers a performance benefit as well. Because the CellDrop is using very flat precision sapphire surfaces and a high precision motor screw to control the arm, the height, there's less variability in CellDrop from measurement to measurement than you see with a traditional plastic slide or a hemocytometer. So yes, it's green and that's great, but it also offers superior performance. So it's a win-win for you in that respect. The CellDrop, using those sapphire surfaces I mentioned, has incredibly clear optics. So you can see an example image off to the side there that highlights our dual fluorescent and bright field imaging all together. So that dual fluorescence I'll cover a little more in a minute, but it offers very powerful analysis in a very fast manner. And the CellDrop also has a ton of modern conveniences you'd expect on any modern lab hardware. So you have your exporting and your touch screen here to integrate into your lab. So a little more about direct pipette. You can see the graphic off to my side here is showing the sample. It's very similar to a hemocytometer, right? I sometimes say it's a hemocytometer with a touch screen. So if you've ever loaded sample in one of those before, it's the same process. You simply pipette 10 microliters of sample between the two surfaces. So you'll load your sample as I just showed you. Then you'll press the count button to analyze your sample. And once the count has completed, you wipe clean with a simple Kim wipe from your lab. And that's it. That's the whole analysis procedure on the CellDrop. So in general, this takes less than a minute and allows you to quickly and precisely quantify a large number of samples so that you can move on to your downstream applications with confidence that you have quality samples to put into the sequencer. So with our samples prepped and ready to go for quality control, I would like to cover a few more features of the CellDrop. So I mentioned that there's a lot of features in our software. The counting protocol is a big one. So the CellDrop offers a variety of parameters that can be tuned from sample to sample. I would like to point out that for the most part, our default protocol works very well with most samples we've worked with, but it's nice to be able to customize any protocol to fit your exact needs. So we know that we can't possibly test every sample type, and we give you the tools to customize it for your needs. So you see here, there's a list of parameters that you can change to make sure it counts according to your expectations. The other criteria of note in the chamber height heading on that protocol screen, you see that we're using a 100 micron, that's standard height it's marked. That's what you're used to if you've ever used a hemocytometer before. That's a 100 micron height. That's a 10 microliter total analysis volume. But the CellDrop offers a 50 micron volume. Because we're not using a fixed plastic, we can adjust the volume using that high precision motor screw I discussed earlier. So as long as your sample isn't super sparse, you can take advantage of this 50 micron chamber height, where you can load as little as 2.5 microliters of nuclei combined with 2.5 microliters of AOPI. Now we know in practice it should be slightly more to build in some cushion for our pipetting, but you can use a very, very small volume of your overall nuclei isolation and save as much for your downstream applications as possible. Once you've customized the protocol, these are easy to set, save, and reload amongst the lab, so it increases precision across the lab by allowing everyone to use the same count parameters. And speaking of all these count parameters, we do have a page on our website, which I'm highlighting here, that has a variety of different protocols that we've worked with scientists in our community, as well as maybe developed in our lab, to provide different starting points for different tissues. So if you work with lung, even if it's not rat lung, this protocol that is on the screen here might be a great starting point. Similarly, the tomato root protocol might be a great starting point if you work with other kinds of plants. So we have protocols on here. This list is not intended to be exhaustive. We always want to collaborate with more people and add more protocols to this page to give even more people tools to analyze their software. But this also brings me to another key point of DeNovix, of what we offer for you, is the application science team is here and ready to work with you and happy to help generate these protocols and tune samples with you as needed, and provide advice and guidance to make sure that your quality control analysis for your nuclei isn't what's holding you back. So we're very responsive and glad to work with anybody that has questions. So now that we've kind of discussed the protocol and you have a bit of an understanding about that, let's talk about the actual imaging. So what the actual analysis process here. So manual nuclei counting with a traditional hemocytometer would look something like this. This was a sample that we imaged here in our lab. This is I think a mouse kidney sample. And you see that there's a lot of dark objects on the screen, and that's the trypan blue stained nuclei. So traditionally, trypan blue would stain dead cells because they have a compromised cell membrane that allows the large dye to pass into the cell. We have removed the cell membrane in the nuclei isolation, so all the nuclei should stain dark. This has been a great analysis tool for a long time, and it's been used for lots of clean tissue culture samples, and it does work really well. The dye is excluded by live cells and goes into dead cells, as I mentioned. But some problems that we know about with trypan blue are that it crystallizes over time. So you can see a few of those crystals in the image on the screen here. But the downside is if you have a really precious volume of nuclei and you load a bunch of it onto your hemocytometer, and maybe your trypan blue isn't super clean, well, it's too late. You've now wasted a bunch of nuclei, and there's a bunch of debris that's confounding your analysis. The other problem that we've run into with trypan blue when counting cells is that it's unable to discern live cells from RBCs. So as I said, live cells with an intact membrane exclude the dye. Red blood cells would have an intact membrane, but we usually don't want to count them. Now, I don't know how many will make it through a nuclei isolation protocol, but they could be there, and you don't want them to be, affecting your extraction efficiency data in an unintended manner. The other thing to point out is that any debris from your extraction, so some of these tissues will have a lot of debris from the actual grinding process, even after filtration. A lot of that debris will appear dark and look like a dead cell. Even to some trained users, it's hard to discern, is that dark object that's circular a dead cell, or is that some debris left behind from my extraction? So those are all challenges presented by counting with trypan blue. And that is one of the reasons that we push the AOPI for counting nuclei. So a background on AOPI, and you can see the nice cartoon on the screen highlighting what I'm discussing here. AO, acridine orange, and PI, propydium iodide, are both fluorescent nucleic acid dyes. So AO is actively transported into live cells and fluoresces green. And PI behaves similarly to trypan blue in that live cells exclude the dye due to its large size, but cells with a compromised cell membrane undergoing apoptosis will include the dye into the nucleus and fluoresce red. So both stains require nucleic acid to bind with to fluoresce either green for AO or red for PI. So that's for cells, but if you flip it for nuclei, what we're talking about is, like I mentioned before, we've removed the cell membrane. So anything that's been any nucleus that's been extracted from a cell should light up red from the PI, and any cell that was not properly lysed in our extraction will stain green. Now, you see on the right that we have AO and PI in any dead cell, which makes sense, but we don't have to worry about compensating or any other complex balancing for the green or AO signal, because there's a fret interaction between AO and PI where the green from AO is quenched through fret by PI. So we have a binary choice of green or red. Isolated nuclei or red, intact cells are green. There's no in between, there's no crosstalk. So instead of viability, we're going to give you an extraction efficiency number using those parameters. And notably, I'd like to point out that I've emphasized nucleus and binding DNA. What that means is any object that does not have a nucleus, so a red blood cell or any debris left behind from your extraction, as you see here, there's a lot of debris in that example image. None of it is fluorescent because it does not have a nucleus because it's not a cell. So we ignore it automatically just using these two stains. So it's a really powerful analysis tool that takes a lot of subjectivity out of the cell counting process. So I'd like to pause for a poll question before we go any further. And I'd like to get a feel for people who are attending. What are your downstream applications? What are you going to do with these nuclei once you've isolated them? Are you going to do ATAC-seq, RNA-seq, flow cytometry? Is there something we haven't considered? If you wouldn't mind entering that into the panel questions, it'd be nice to know what other techniques are out there that people are using for these isolated nuclei. I do see some results still coming in, so I'll give everyone a few more seconds here to answer. But looks right now like we have a landslide winner. I'll reveal in a minute, but I think everything's kind of halted here. So let's end the poll. And the majority of you seem to be doing RNA sequencing, but we do have a good number of people that are doing flow cytometry or ATAC-seq with their isolated nuclei. So the preference seems to be RNA, but we have some other representation here as well, and a little bit of other. So again, I'd like to know what that other really is. So if you wouldn't mind telling us, that would be great. So now we've kind of talked about the imaging and the counting. Let's actually move on to a demo to highlight how the CellDrop can most effectively count and quantify these nuclei. So I will step over to my demo instrument, my CellDrop here in the lab. And I'm going to work on resuspending my nuclei. So you can see here on the screen that we have a number of applications that the CellDrop can use to count cells. We can use the CellDrop for AOPI quantifications of tissue culture or cell isolations. We have tryptophan blue counting methods. We have even GFP methods, but today we're going to focus on this Nuclei AOPI application. So you simply tap it to load it, and this is our live preview screen. So you also, now I need to load my nuclei. So while I was talking, I was resuspending my sample. I have 10 microliters in my pipette, and I'm going to, just like the cartoon showed a minute ago, put the pipette tip in the groove and pipette it onto the CellDrop. So these nuclei were isolated today, and you can tell we had a really good isolation. We have a lot of cells here, or a lot of nuclei, excuse me, on the screen here. And we can pinch to zoom, we can look around, we can just check on the sample as it's moving. So we can see that it's still settling, and we want to give it a few seconds to do that since we are going to take multiple images and overlay them. In the meantime, it's a good idea to name your sample. Everything on the CellDrop is automatically saved, so naming it will help you keep track of it later. And the first thing I would recommend doing is changing over to our red fluorescent channel, our PI, or what we expect to see, or where we expect to see our nuclei. Since this is where a majority of our sample will be, we are going to use this channel for focus and exposure. Now, I used the CellDrop earlier today to quantify this sample, make sure we had enough for the webinar, and you can see that it is still in the correct focus position, and it's still bright enough. I wouldn't change the exposure or the focus here. So you see that it's not necessary to do that every single time, unlike something that requires a plastic consumable or even a hemocytometer. There is variability between every single slide. In this case, we're not using those slides, so we don't have that variability problem. Once it's focused and exposed, it generally needs very little tuning. But if you wanted to make those adjustments, our focus button here, we have autofocus. We also have manual focus, so if you need to just tune it a little. And we also provide guides. So if you're not sure how to focus, we should give you some hints. Similarly, we want to make sure that everything's bright enough. If you've ever done any kind of fluorescent imaging, you know that you want to minimize background signal. So you want to have as little noise in the background as possible, but you want to have as much signal on the sample as possible without oversaturating. Again, I think our exposure is fine. I'm not going to tune that at all, but we provide guides again if you're not sure of what to do. Considering all that, we're going to move over and check our green exposure. I don't expect to see a lot here because we're expecting our isolations to go well. I see maybe one object. That's good. I don't want to make any more adjustments here because it'll affect every other channel. So I'll just return to my bright field image, check that nothing's moving, which it's not. I will use the default protocol, which, as I mentioned, is good for most samples, and I will press count. So our count process should take around 10 seconds. It takes 3 images, a bright field, a green, and a red image. It overlays them and counts them. So in this case, our red image will be used for approximating our nuclei or counting our nuclei. Our green image will be used for counting any cells that didn't make it or didn't quite get lysed in our extraction. And our bright field image isn't counted. It's just there for reference so we can see the morphology of the objects we're counting. Now you can see, again, I mentioned earlier, we had a really good extraction. So we are seeing 28 million nuclei, which is great. That would require probably some heavy dilutions before we go off to sequencing, but it's a good problem to have today. We have 100% extraction efficiency, which is also, fantastic to see. And we have 5,000 events counted. So you can see here the nuclei plus cells. We have 5,000 total events on the screen, which is statistically very representative of what would be in the tube. Using the cells per mil and the nuclei per mil numbers, we can calculate an extraction efficiency of 100%, like I said. And right here at the bottom, the number everyone probably cares most about is our 28 million nuclei per mil. Now, you can zoom in on this image to just see how it was counted and make sure that you agree with everything. But it does generally look very good. You can gate by size. So you click that little histogram, you can show the distribution of cells per micron diameter. And if you wanted to truncate that further to exclude anything that you don't want in your analysis or extend it, depending on your application, you can do that. And let's say you wanted to maybe tune this protocol. So let's say you were zooming in and looking around, and there was a substantial population of cells or nuclei that you wouldn't have counted the way they were counted. We offer this optimize settings button, which this should look familiar from a previous slide. You can go through and make some adjustments here and then apply that new math to the images on the previous screen. We're not going to take a new image. We'll just reprocess with different math. And we'll do our best to work with you here to tune the protocol to best count the cells or the sample the way you would count the sample. Once you've made those adjustments, you'll be presented with an option to save that protocol. So once you've done the hard work of tuning it, you can save it and load it later, or anyone else in your lab can use the protocol that you have tuned to count your sample later or count the same sample another day. So you always have access to the same exact settings and you can use them over and over again, which is great for standardizing protocols across the lab over the duration of an experiment. Another important consideration in nuclei isolations is how many clusters or clumps of nuclei you have. So the CellDrop can also help you visualize that. Right now you see we're in the single sample view. If you change to the graphs view, you are presented with, here's your extraction efficiency graph, but we want to change it to cluster size. So this will show us how many clusters and how many cells are in that cluster on the screen. So you can see here that we have 2,553 clusters. Really wouldn't be the right word because it's one cell. So it's of individual cells, we have 2,553 of those. And then we have 636 double cells. So it's two cells that are stuck together, but we can tell that they're two cells. So we've counted them as individual cells, but we're telling you they're stuck together and touching, which this can cause some downstream issues during your library prep. So this is a great graph that enables you to make decisions about the quality of your sample before you move on to the actual library prep phase. If you had a really high number of clusters and a really low number of single cells, you may want to look into other options to better homogenize that sample and reduce the number of clusters in there. But you do get that tool right here to analyze it quickly on the CellDrop. And then lastly, you might want to know how you can access this data later on your computer or show your PI or show some colleagues. You can click this export button here and you're presented with a list of options. You can get a results CSV. So this will just be something you can open in an Excel file. You get a PDF report where you can include images and histograms like I just showed you. And you also get access to the raw images. So these are just exported as .png files. They're easy to access on any computer. And you can do any other downstream analysis that you may want to do using those images. So that's all, those are all options you have here on the CellDrop. And when you're finally done analyzing your sample, you would just hit next count. So we'll return to our live preview screen, and I'll show you how to quickly clean the sample surfaces. So it's quite an easy protocol. So you simply lift the arm, and then using a dry, clean Kim wipe, you apply firm pressure to both surfaces. And when I lower the arm again, you can see that the live preview is quite clean. Any small amounts of debris like that can be flushed out with ethanol, but also are generally flushed out by the next sample. So in this scenario, all of this would be flushed away by the next sample. So that, we're ready to go for our next sample. And if I wasn't doing a demo, we probably could have processed 10 samples by now. So you can see that the CellDrop counting process is very efficient and very quick and allows you to get a lot of powerful analysis in a fast manner to move on with your actual downstream application. So with our live demo complete and with our nuclei isolated, quantified, and analyzed for any other considerations before you move on to your library prep, we want to highlight a couple of other options here. So of course, we had a nice sample to cooperate for the webinar. We want to highlight some images that we have generated or some of our customers have generated that show all the different facets of nuclei isolation protocols. So we'll start with this first image here. This is fresh kidney, so not frozen, not FFPE preserved. You can see that there is a good amount of debris in the sample, but not too much. That's another thing that the CellDrop allows you to do in real time when you're looking through the image or when you're looking at these images. You can analyze visually for debris and say, there's too much in there, I need to do further filtration or purification before I can move on with library prep. Or in this case, maybe you're happy that level of debris isn't too bad. You can see that we have a nice distribution of single nuclei and very few intact cells. And that was using this 10x genomics kit. Similarly, we did a frozen kidney, and this was using the Miltenyi GentleMax system and their extraction buffers. You see a very similar level of debris. Again, I don't think this is too much debris to move on. And this level of the nuclei are mostly single. There's very few nuclei that are stuck together and very few intact cells. So this was another extraction that proceeded very well. And you can quickly, within a minute, make all these decisions about this sample and move on to your downstream application. This was a frozen lung sample that we processed here using our homebrew buffers, similar to the buffers I used for today's extraction. Again, these are just generated using some protocols from the literature. The next sample is from one of our botanists, and that is a fresh tomato root sample. This was one of their homebrew kits. This is something that they came up with to extract nuclei. Again, you can see that there is some debris, but not a ton. Using the images, very few intact cells. And even though there are a variety of shapes of nuclei on this sample, they're not all perfectly round. We do a really good job of recognizing the individual nuclei or the clustered nuclei and counting them appropriately for you. And finally, we'll show a frozen mouse brain sample, which brain samples in general have just been the most debris-laden samples that we have. You can see a large amount of background debris in the sample. And you do see a good number of isolated individual nuclei. I don't see any clumps of nuclei, but the level of debris is such that I probably wouldn't want to move on with my library prep until I've done further purification, because that's going to get in the way on the microfluidic. You may even clog your microfluidic by loading this much debris into it. So before we move on, we can use a purification kit to just purify out the nuclei. So Miltenyi offers a anti-nucleus microbead purification kit that takes advantage of magnetic microbeads that are quite small and allows you to essentially wash away all your intact cells and debris. So this same sample, this is the same sample. It might be hard to believe it, but this image that just kind of moved onto the screen is the exact same sample after we ran it through the purification kit. So you can see that it's highly effective in removing debris from the isolation, as well as any maybe intact cells that were left behind or other clumps. And it doesn't affect our yield. There's still plenty of nuclei on the screen. There's still a good concentration. It's just our extraction efficiency has been improved by purifying with this kit. If you want more information about the ins and outs of how this kit works and the exact specifics of how to use it, when to use it, all those things, I would recommend going to the Miltenyi website where you can view that protocol there. I just wanted to say that we recommend it when you have a messy sample like this. So with our live demo complete, our nuclei isolated, purified, all of those considerations, we're happy with our sample. We're happy that it is a high quality sample based on all the analysis we've been able to do on the CellDrop, and we'll move to our downstream applications. So it seems like today the popular choice is RNA sequencing, but ATAC sequencing and flow cytometry are also other options here, and they all require a sample with plenty of nuclei, very little debris, very little intact cells. And you want your nuclei to be at a good concentration before you load them onto that chip as well. So you'll know the exact concentration of nuclei that you have, as well as the level of debris and intact cells in that sample. So you can be confident that when you go load them into these downstream applications, that it's a good sample. It's a quality sample that will hopefully give you good results in those applications. So in summary, I'd like to bring our attention back to what the CellDrop offers here. These nuclei extractions are lengthy, sensitive, and they're expensive procedures. There's a lot of equipment that we've talked about today. There's lots of kits and filters, and there's a lot that goes into these. And that's just to get to the nuclei isolation. That's not even to process those nuclei with RNA-seq or ATAC-seq. Some of those runs are upwards of $5,000 or $10,000. So you want high quality samples before you go off and run one of those experiments. Sample QC, sample quality control, is a critical component to ensure that you're not wasting money on a sample that wasn't extracted well or had some other issues to it that can be easily visualized by simply counting. So with image-based counting on the CellDrop, we're able to reproducibly QC all your nuclei samples in a way that is easy to interpret and powerful, that allows you to make quick decisions before you go on with your experiment. I'd like to also draw your attention to some additional resources. It's always impossible to cover all the information about something like this. So please visit DeNovix.com. We have a series of ebooks. We have other webinars that cover other counting methods and counting protocols on the CellDrop. We have plenty of technical notes about different applications, including different nuclei isolation applications. And we have plenty more, like you saw our recommended protocol screen. We have so much to talk about in videos, so much to talk about that I can't cover it all even in one slide. So I would recommend visiting the website for any other information you would like about the CellDrop. And in fact, that QR code that you see on my screen right now can take you to all this information. So I'd recommend either screenshotting this slide or taking a picture with your phone so that you can go back and access this wealth of resources later. Most importantly, I'd like to encourage you to schedule a virtual demo or a free trial with myself or someone else on the application science team so that you can see how the CellDrop might improve your nuclei extraction workflow. So we can do something similar where myself or another member of the team hops on Zoom with you in a personal session, and we can work one-on-one with directly addressing your sample and your specific questions that apply to your workflow. Or more importantly, the best way to know if something works for you is to actually try it yourself. So we do a free trial program in the US where you can sign up for the free trial. We will mail you the device. We cover the shipping to you and back from your site. And during that free trial, you can work with myself. You can work with another member of the application science team to ask any questions, tune, optimize the equipment for your specific workflow and your specific samples. And then once you've been satisfied with your ability to count and quality control your sample on the CellDrop, you simply pack it up and mail it back to us when you're done. So that's also available through that screenshot or through that QR code on the screen here. And with that, I'd like to take questions from the audience that have come up. I've seen plenty of questions flying by here, and I believe the team has been hard at work answering them, but I'd like to take any relevant questions that I can in the time that we have left.
Thanks, Ben. Yeah, you're right. There were a lot of questions came in through the course of the presentation. So I'll just put a couple of these out to you and to the other guys, Dan and Dave, on the call at the moment. First question I have is about irregular shaped nuclei. Basically, how does CellDrop deal with irregular shaped nuclei?
Oh, it's a great question. So the CellDrop and a lot of other automated cell counters are designed to count circles. So a lot of tissue culture cells, PBMCs, all your classical kind of reasons you would want to count, they're circular. But we have some additional modifications in our algorithm. It's specifically called irregular cell mode. There's a technical note on our website about how it works. But in short, you can turn that on. You probably saw it on those protocol screens I was showing you. You check the box to turn on irregular cell mode, and that allows our algorithm to better process oval or oblong shaped nuclei, which are common in certain samples that contain smooth muscle. Smooth muscle nuclei are known to be almost rod shaped and in how they appear. So things like the heart is a good example where we see a lot of these, but you can turn on this irregular cell mode that will allow you to better process those ovals.
So our second question is, we image our nuclei on a microscope after counting. Is it possible to use slides with CellDrop so we don't need to use additional sample for this?
Yeah, it's a great point. So I mentioned dropping the arm down to use the smallest amount of volume possible when counting our nuclei. And the same way that we can lower the arm to that lower height and use less volume, we can actually raise the arm up and accommodate these plastic slides. So the CellDrop arm will move up substantially from where it currently is, and it will leave enough space for a plastic slide. Once you have loaded your sample into the plastic slide, the whole procedure is the same as everything I showed you. You put it over the camera, count, and then when you're done, you can take that off to a high-powered microscope, probably 40 or 60x objective, and do your nuclear envelope integrity analysis without doing wasting too much sample and quality control.
Right. This is a question that maybe Dan or Dave can jump in on if you don't have the figure to hand. But what is the recommended concentration when counting using AOPI if we're making it ourselves?
I don't have the exact micromolar concentrations of our dye on hand, on top of my brain. It is on our website. So if you go to our page where the AOPI is listed, you can find those concentrations and make it yourself. It's nothing overly crazy. The nice thing about buying it from us is it's already quality controlled here heavily. We do a lot of analysis before we send it out to make sure it is within very tight tolerances to be mixed 1-to-1 and ready to go for imaging on the CellDrop. So there are advantages and disadvantages to making your own, but the concentrations are on our website somewhere, or Dan and Dave maybe could chip in if they have it.
Yeah, I'll jump in. So what we have in the tube, if you were to buy it from us, you would be getting 5 microgram per mil AO and 100 microgram per mil of PI, and that's in PBS.
Thanks, Dan.
Thanks. Another question's come in. Using CellDrop, how do the results compare to using a hemocytometer or a flow cytometer?
So with a hemocytometer, as I've pointed out from my slide, there's a lot of subjectivity in those counts. So the CellDrop tends to align closely with maybe the average answer over a large, 10 replicate study, but the precision of the CellDrop is much tighter than the hemocytometer. So you'll see a large variation from count to count in the different hand counts, just because of different users and having to do so much interpretation of the image ourselves. The CellDrop still gets a really accurate count, but it's far more precise than what the hemocytometer can offer. And now, in terms of a flow cytometer, we have some data that shows we are very closely aligned with the flow cytometer we've worked with. So there was no statistical difference between the counts on the CellDrop versus the flow cytometer that we were able to get our hands on. So we do compare favorably with both technologies, just a little differently.
Can you differentiate nuclei from dead cells?
No, we cannot by AOPI. We can via size. So the dead cell would be a lot larger than an extracted nucleus. But it's also highly unlikely that a cell that is dead made it all the way through the extraction process and didn't, I guess, lyse the nucleus out. So there's a statistically low chance that a lot of dead cells made it all the way through the washing, all the filtration. There's, that montage I showed was just that. It was a montage. It was not the full protocol. If you want to get a picture for what the full protocol looks like, I recommend going to Miltenyi's website or another manufacturer's website and reading the full details. There's so many washing, filtration, spinning steps that I would be pretty surprised if a huge population of dead cells made it all the way through to that extraction. But again, you could gate via size in the red channel to eliminate any really large dead objects. That would probably eliminate any small number of dead cells that make it through.
There was a follow-up question to that as well, that if you cannot determine between dead cells and isolated nuclei, would you recommend doing a viability count pre-isolation and working out from there the live dead isolation efficiency?
You could. The other probably more simple way to do it is another kit. There's a dead cell and debris extraction kit, I think, from Miltenyi. So that could help purify out a lot of that stuff as well. But yeah, that's another option.
Thanks, Ben. Another question that we had in was specifically for nuclei. Can't we just use AO or PI? Do we need to use both?
You could use either. You could use one or the other. If you use just AO, you wouldn't have any idea of efficiency. So it would stain your nuclei, but it would also stain your live cells. So you wouldn't know which objects are live cells intact that weren't lysed by the extraction and which are nuclei. You would hope that they're mostly nuclei, but you won't have any idea if your extraction really went well, let's say. And similarly for PI, it'll do fine staining just nuclei, but you won't have a super strong idea of how many live cells are in your mixture. And knowing how much debris and how many other particles of interest, if you will, are present is going to be critical for a high quality library prep. You want to make sure that there's nothing taking up space on the microfluidic when you go to process for the single cell sequencing.
These are very low cost assays as well, so there isn't a tremendous amount of benefit in just using one of the fluorophores.
Correct. That's another worthy consideration. They're not antibody pricing. They're pretty easy to buy in bulk for relatively cheap. So I would say the more powerful analysis is the combination of AO plus PI, and it's not terribly more expensive than the combination isn't terribly more expensive than one single dye.
Okay, it's kind of an interesting one. So you have applications for AOPI, nuclei AOPI, and primary AOPI on your home screen. Mm-hmm. What is the difference?
So the only difference between the regular AOPI and the primary AOPI are the default settings. So we wanted to have default settings that would get you closer to your expected cell count when you use primary cells versus tissue culture cells. So we saw there was a lot of additional considerations that had to be made for primary tissue, specifically things like RBC autofluorescence. If you aren't really careful about tuning your exposure, we had a lot of people that were overexposing heavily in the red channel. And what that would do is overestimate their dead cells. And that was just due to the red blood cells having some autofluorescence due to the heme groups. So we have settings that are tuned to give you better results from the get-go as the default when you do a primary cell workflow versus a tissue culture cell, which is the standard AOPI application. So there is no difference functionally in how they count. It's just the default settings that they use.
And a question sort of related to that is, should we focus in the red channel for nuclei and focus in the green channel for standard apps? And can you explain why this is?
Yes, that's correct. And the reason you would want to do that is, I kind of alluded to this when I was talking earlier, but in the Nuclei app, when we were focusing in the red channel, we expect a majority of our population, of our sample, to be in that red channel. We expect that our isolation went well. We have no reason to suspect otherwise. So we would want to focus in the channel where we have the most spread evenly across the screen. So we want to have the ability to focus and expose where we have a large number of events all across the screen to make sure that the exposure and focus we pick is applicable for the whole sample we're counting. We don't want to do that in somewhere where there's very few events. Similarly, when we're doing an AOPI count, for the most part, people want high viability counts. They don't have really dead tissue culture cells. So it's pretty hard to, do that bad of a job with cell culture. So we expect that a majority of our cells in AOPI are going to be green when you're counting tissue culture cells or tissue extraction. And therefore, similar principle, you want to focus and expose where you have the most representation on the screen.
Okay, great. What is the minimum amount of nuclei sample that you recommend using? I think you touched on this earlier when you were talking about chamber heights.
Yeah. So the minimum amount of sample we would recommend using is, for that 50 micron chamber height, is 2.5 microliters, 2.5 microliters of your nuclei solution and 2.5 microliters of your AOPI. Mix them together for a total of 5 microliters to load into the 50 micron height. Again, I think I mentioned earlier, in practice, it would be a little better to, account for pipetting error and probably do 3 microliters and 3 microliters. But you can get away with as little as 2.5 and 2.5 if you're really careful about your pipetting, to minimize how much of your actual extraction you need to use.
Thanks. Do you recommend using wide bore tips for mixing and loading?
There are several steps in the Miltenyi protocol that recommend wide bore, and in our own protocol that recommend wide bore, specifically our own. I know for a fact the homebrew buffer we used, I want to double check the Miltenyi thing before I say something that's not true. In our own homebrew kit, the one I did today, we use wide bore tips for some of it, but it doesn't seem to be necessary for everything. I used a standard P200, yellow tip during the extraction here, or during the loading process here, and I didn't have any issue with loading all the nuclei that you saw on the screen there. So I don't think it's necessary for everything, and definitely not necessary for loading on the CellDrop.
And there's a question just came in as well on what are the concentration ranges for CellDrop? And again, that ties into the chamber heights that you were discussing.
Yes, that's exactly it. So the reason we have those chamber heights is to accommodate a really wide variety of concentrations or cell densities. So the 50 micron height is best for really dense samples or really concentrated samples. For the most part, we recommend using that upwards of 10 million cells per mil. Anything under that and down to about 100,000 cells per mil, we recommend the 100 micron height. And then from 100,000 cells per mil to about 700 cells per mil, we recommend the 400 micron chamber height. So that's 4 times as high as your standard heme cytometer. So instead of 10 microliters there, you would load 40 microliters, which might be less than ideal for a nuclei sample. But those numbers are just based on, statistical significance. You can certainly push them, and there's nothing in the CellDrop. There's no error code or nothing that prevents you from using things outside of those ranges. It's just what we've characterized as being linear and cooperating nicely. For the nuclei, if you're less than 10 million per mil, which probably many extractions will be, you can still get away with that 50 micron chamber height. So if you have questions or are curious about the exact densities or your images and whether the 100 or 50 micron height is a better option for you, definitely send some examples and to the application science team. We'll be happy to work with you on that.
Thanks, Ben. So I think we've answered all of the questions now. We will take a look through all of the questions and check that we've gone through and answered them all, and we'll get back to anybody if we've missed any questions there. But I think we'll leave it at that for the Q&A session now. And I just want to thank you, Ben, for your presentation and for everybody for attending. The presentation itself is based loosely on this infographic that we have available. So this is available to download from our website, but also you'll get a survey after the webinar, and you have the option to request a hard copy of this if you'd like one to hang up in your lab. So as I mentioned, you'll get a survey. Hopefully you'll fill that in. It gives us sort of useful feedback and information about how we're doing and what you'd like to see in future webinars. And we'll also make a recording of this available for you either this week or early next week, and we'll send that to you in a separate email. You can get loads more information on DeNovix.com, and as Ben mentioned, you can request a virtual demo or a free trial or quotations on our website. For those of you who are joining us from outside of the US, we do have a full distribution network globally, and all our distributors are fully trained with CellDrop and DS11, and they'll be happy to come and do a demo in your labs or arrange a free trial for you as well. But I think I don't think there are any more questions at the moment. So all it leaves us to say is thank you very much, Ben, and thank you everybody for attending our webinar today.
Thanks, Andy. Thanks, everybody.